Dear friends, good morning. Today we can discuss structure, functions of coenzymes and isoenzymes. Structure and functions of coenzyme. Definition: a non-protein organic substance loosely attached to the enzyme and can be separated by dialysis and is essential for enzymatic action. It is also called as co-substrate or coenzyme. The coenzyme is often derived from a vitamin, for example, NAD, FAD, thymine, pyrophosphate. Characteristic features of coenzymes: they are stable towards heat, they are generally derived from vitamins, they function as co-substrate, they participate in electron transfer reactions, example NAD+, group transfer reactions, coenzyme A. The table shows the functions of some coenzymes: NAD plus FAD, FMN, which involved the hydrogen transfer reactions, example proton or electron. Thymine pyrophosphate, which involves the acetyl group transfer reactions, CH3CO. PLP, pyridoxal phosphate, which involves the amino group transfer reactions, NH2. Biotin, which involves the carboxyl group transfer reactions, example COO minus as a carbon dioxide form. Coenzyme A, acetyl group transfer reactions, or CO. THF, tetrahydrofolate, which involves the transfer of carbon 1 unit or 1 carbon unit, example methyl or CH4 group. Lipoic acid, which transfer reactions similar to biotin, example carbon dioxide or CO minus. Now we can discuss deeply in each coenzymes. First one, nicotinamide NAD or NADP plus as a coenzyme. Sources for NAD: vitamin niacin, vitamin B3. This is the structure of NAD as well as NADH. First one is for NAD, either phosphorylated, dephosphorylated form. Second one is NADH, reduced form of niacin. The functions of NAD, NADP plus. NAD, nicotinamide adenine dinucleotide, NADP, nicotinamide adenine dinucleotide phosphate. These coenzymes play an important role as hydrogen and electron transfer agents by means of reversible oxidation and reduction. The mechanism of transfer of hydrogen from substrate or metabolite to oxidized NAD, causing the oxidation of the metabolite or substrate and the formation of the reduced NAD+. Examples of NAD catalyzed reaction. First one is conversion of lactate to pyruvate. Lactate reacts with NAD plus in the presence of lactate dehydrogenase enzyme to form the pyruvate and reduced form of NADH plus. Second one example, conversion of glucose 6-phosphate to 6-phosphogluconolactone. Third one example is conversion of dihydrofolate to tetrahydrofolate. NADPH H plus reacts with dihydrofolic acid to gives the NADP and tetrahydrofolate. Fourth one example is conversion of pyruvate to acetyl-CoA. Pyruvate reacts with NAD in the presence of pyruvate dehydrogenase to form the acetyl-CoA and NADPH H plus. Fifth one example is in electron transfer chain, either of these coenzymes accept hydrogen from the substrate and then transfer it step to other compounds. Substrate to NADP, then FAD, then coenzyme Q, then cytochrome B, cytochrome C1, cytochrome C, cytochrome A, cytochrome A3. Finally, we can get the half molecules of oxygen. In this way, NAD or NADP function as coenzyme for metabolism of carbohydrate or lipids or amino acids and keto acids. Next one example, FMN or FAD as a coenzyme. Sources for FAD or FMN, riboflavin, vitamin B2. Structure of FAD, FMN. These are two flavin containing coenzymes synthesized from vitamin riboflavin in our body. Functional part of enzyme is isoalexin ring of riboflavin. It is involved in oxidation reduction reactions. FMN means flavin mononucleotide, FAD means flavin adenine dinucleotide. This is the structure of FMN, FAD. First one is flavin mononucleotide, second one is flavin adenine dinucleotide. Functions of FMN as well as FAD. The general reactions of FMN FAD catalyst reaction. AH2 as a one substrate reacts with FAD, which gives the A, and hydrogen accepts the FAD to form the reduced form of FADH2. The reduced form of FADH2 then reacts with the second substrate to form the reduced form of second substrate converted to product, then oxidized form of FAD. First one example, dehydration of succinate to fumarate in the citric acid cycle. Succinate reacts with oxidized form of FAD in the presence of succinate dehydrogenase enzyme to form trans fumaric acid or fumarate plus FADH2. Second one example, pyruvate dehydrogenase complex also requires FAD as coenzymes for conversion of pyruvate to acetyl-CoA. Third one, FAD as a coenzyme D-amino acid converted into the corresponding keto acids. On the other hand, L-amino acids are oxidized to keto acids by enzyme containing FMN. D-amino acids only reacts with FAD in the presence of D-amino acid oxidase, which produces the corresponding keto acid plus FADH2. L-amino acids only react with flavin mononucleotide in the presence of L-amino acid oxidases, which gives the corresponding keto acid plus FMNH2. Fourth one example is acetyl-CoA reacts with FAD in the presence of acetyl-CoA dehydrogenase enzyme, which produces the alpha-beta unsaturated acyl-CoA. Fifth one example, xanthine in the presence of FAD, xanthine oxidase enzyme to form the uric acid. Next one, coenzyme A, otherwise called as CoA. Sources for coenzyme A, pantothenic acid or vitamin B5. Structure of coenzyme A usually referred to as CoA for short form, but it is written in the equation as CoA-SH because it has thiol group as the reactive part of molecule. Coenzyme A widely involved in the transfer of acyl groups. The terminal sulfhydryl groups in CoA is reactive site. Acyl group are linked to CoA by thioester bond. This is the structure of coenzyme A, which consists of acetyl group, pantothenic acid and phosphorylated adenosine diphosphate. Next one, functions of coenzyme A, which helps in the transfer and activation reactions involved acyl groups. First one example, conversion of palmitate to palmityl-CoA. This is the active form of fatty acid. Palmitate reacts with coenzyme A or CoA-SH in the presence of ATP thiokinase enzyme, which catalyzes this reaction, which converts palmityl-CoA and AMP adenosine monophosphate plus pyrophosphate PPI. Second one example, conversion of pyruvate to acetyl-CoA. Here pyruvate reacts with NAD FAD coenzyme A in the presence of pyruvate dehydrogenase enzyme to produces the acetyl-CoA carbon dioxide reduced form of NADH and FADH2. This reaction acts as a link between glycolysis and also TCA cycle. Third one example, conversion of acetate to acetyl-CoA. Acetate reacts with ATP and CoA-SH in the presence of thiokinase enzyme, which produces the acetyl-CoA and AMP inorganic phosphate. Coenzyme A combines with acetate to form active form of acetate, that is acetyl-CoA, which is directly utilized by combination with oxaloacetate to form citrate, which initiates the citric acid cycle, TCA cycle. Fourth one example, transfer of acyl group. Condensation in which the acyl group is attached to a nucleophilic resulting in release of coenzyme A. Choline reacts with acetyl group which gives the acetylcholine and also CoA. Fifth one example, in lipid metabolism, coenzyme A has got significant one. Acetyl-CoA forms ketone bodies during oxidation of fatty acids. Acetyl-CoA is the precursor for cholesterol biosynthesis. It is the main source for synthesis of cholesterol as well as steroid hormones. Sixth one example, sulfonamide drugs are detoxified by acetylation with acetyl-CoA. Next one, coenzyme, biotin. This is a prosthetic group which strongly binds with enzyme. Enzyme source, NAD vitamin source B7. Structure of biotin, the structure shows the structure. Function, biotin acts as a coenzyme in the fixation of carbon dioxide in carboxylation reactions. First one example, pyruvate reacts with biotin carbon dioxide in the presence of ATP in the pyruvate decarboxylase enzyme to form the oxaloacetate. Next one example, it is also act as coenzyme for acetyl-CoA to convert propionate to succinate. Third one example, it involves the fixation of carbon dioxide in purine synthesis. It is required for carboxylation of acetyl-CoA to form malonyl-CoA in the fatty acid biosynthesis. It is also required for carboxylation of propionyl-CoA to methyl-malonyl-CoA. Sixth one example, green plants require it for fixation of carbon dioxide. It is also involved in biological synthesis of oleic acid. Next one example, folate or folic acid, the source of folate, vitamin B9. The structure of vitamin B9 or folate consists of pyridine ring and para-aminobenzoic acid and also glutamic acid. Functions of vitamin folate, the tetrahydrofolate is involved in one carbon metabolism. It serves as acceptor or donor of one carbon units, formyl or methyl group, in a variety of reactions involving amino acid and nucleic acid metabolism. It is involved in serine glycine interconversion catalyzed by enzyme serine hydroxyl methyl transferase. Fourth one, it acts as a coenzyme for conversion of homocysteine to methionine. Fifth one example, it is mainly required for maturation of RBC, thus in tetrahydrofolate megaloblastic anemia is found. Purine and pyrimidine synthesis requires folate as a coenzyme. Next one, lipoic acid. Lipoic acid is a water soluble material containing 8 carbon unit and also 2 sulfur atoms. and hence it also termed as thiocytic acid. It is widely distributed in natural foods, especially yeast, milk, kidney, liver, and also green leaves. This is the structure of lipoic acid. Oxidized form and also reduced form. Function of coenzyme. This coenzyme involved in pyruvate dehydrogenase required along with other coenzymes in oxidation of decarboxylation of pyruvate to acetyl-CoA. Alpha oxoglutarate dehydrogenase complex required along with other coenzyme in oxidative decarboxylation alpha oxoglutarate to succinyl-CoA formation. Lipoic acid is also required for the action of the enzyme sulfate oxidase required for conversion of sulfate to sulfite. Isoxanthin is also required for action. Next one, isoenzymes. Isoenzymes definition: many enzymes are present in more than one molecular form in the same species, in the same tissue, even same cell. Such form of enzymes which catalyze the same reaction but exhibit different kinetic properties are called as isoenzymes or isoenzyme. Isoenzymes or isoenzymes. Physically distinct form of same enzymes are also called as isoenzyme. Based on example, lactate dehydrogenase (LDH). These enzymes catalyze the same reaction but differ in amino acid composition. Arises through gene duplication. Exhibit subtle differences in properties such as sensitive to particular regulatory factors or substrate affinity. Lactate dehydrogenase, it is found in most cells of human body. 5 isoenzymes and these are associated with different tissues, in particular liver and skeletal muscles containing mainly M4 form, while those of heart muscle contain mainly H4 and MH4. All of these isoenzymes found in kidney cells and also erythrocytes RBC. H4 type present in heart muscle, M4 present in liver and also skeletal muscle. All 5 isoenzymes are present in kidney cells. LDH enzyme exists in two forms. One is heart LDH, predominantly present in heart, and another one is muscle LDH, which is predominantly present in muscle. 4 H's HHHH LDH1 which present in heart, HHHM LDH2, HHMM LDH3, HMMM LDH4, 4 M's. This is for LDH5, especially present in muscle. Next one example for isoenzyme, creatine kinase. Creatine kinase, also known as creatine phosphokinase or phosphocreatine kinase enzyme, expressed by various tissues and cell types. In these in tissues and cells that consume ATP rapidly, especially skeletal muscle, but brain, phosphoreceptor cells and of retina, hair cells of inner ear, spermatozoa and smooth muscle, phosphocreatine serve as a energy reservoir for rapid buffering. Thus, creatine kinase is an important enzyme in such tissues. Clinically, creatine kinase is assayed in the blood test as a marker of damage of creatine kinase rich tissues such as myocardial infarction, heart attack, rhabdomyolysis, severe muscle breakdown, muscular dystrophy, autoimmune myositis, and acute kidney injury. The structure shows creatine in the presence of creatine kinase, in the presence of ATP converted as phosphocreatine. In human tissues, creatine phosphokinase exists as 3 different isoenzymes, Each isoenzyme is a dimer composed of two protomers, M for muscle, B for brain. Creatine phosphokinase 1, BB, which expressly present in brain. Creatine phosphokinase 2, MB, which expressly present in myocardium. Third one, CPK3, consists of MM, especially present in skeletal muscle. In addition to above three form, two typical isoenzymes of CPK have been reported. They are macro CK macro, otherwise called as mitochondrial CK, otherwise called as CKMI. Thank you.